Hello everyone, welcome to Barcode Biotechnology. So I, Prakash Rao, welcome you all to the today's workshop entitled AI Assisted Drug Design from the ADMET to pharmacophore modeling. Okay, so before going to the session, I suggest you that you must have your pen and paper ready so that you can write some of the important terminologies and important step-by-step process. Okay, that we are going to discuss about. Okay, so without any delay, let's start the session. Okay, so before going to ADMET prediction and pharmacophore modeling things, first we have to learn why we are studying this. Okay, so first we have to learn about the process of the drug discovery to the drug approval process. Okay, so first of all, the point is target identification. Okay, so if we don't have any purpose in our life, so what is the direction of our life? We have a proper goal. In the same way, we have a proper target. We have to find out what is our target protein, whether it is we are working on bacterial enzyme, whether we are working on any fungal enzyme, or whether we are working on any viral, okay, in virus, or we are working on inflammatory, all those things like COX enzyme, cyclooxygenase. Okay. So for example, we are working on any bacterial enzyme that is the DNA guidase. Okay, so let's suppose we are working on DNA guidase, the antibacterial enzyme. Then the next process that when our target is get fixed, then we go for the heat identification. Okay, so basically heat identification means you find out the this is your bacterial target, we find out or we search for the drug molecule having the similar effect compared to the marketed drugs. Okay, we just screen means we find out different drugs so that we can optimize to a lead compound. Okay, that is, it is a set of 100 to 200 drugs, 100 to 200 drugs having the antibacterial activity. Okay, so this is nothing but some drugs that is having the antibacterial activity. Because if we take the antibacterial drugs, then we can analyze more properly. Okay, so how to prepare this one? We are going to discuss it about that. Okay, after the heat identification, we go for the lead optimization. Okay, so from this 100 to 200 drug antibacterial drug, we perform the molecular docking process. Then we screen out only 2 to 3 compounds. Okay, 2 to 3 compound for the lead molecule. Okay. After that, we perform various studies like we go for the ADMET. We go for the second thing that is the DFT. We perform some sort of analysis, interaction analysis, because we have 100 to 200 heat molecules. That heat molecule, we perform the docking. After performing the docking, we go for various analysis. Then finally, we get our lead molecule. Okay. After that, we optimize that lead compound. Then we go for the fourth step, that is the preclinical trials. So in preclinical trials, this is also called as the microdosing stage. Okay, I can write it here also in down the microdosing phase. Okay, this is the microdosing phase. This in this microdosing stage, we test our drug molecule on the animals such as rat, such as the rabbit, the guinea pig, okay, all those animals we study. After successful in vitro study, or you can say the preclinical trials, we go for the clinical trials where we have the 3 phases, okay? We have 3 phases of clinical trials. That is the phase 1, phase 2, and phase 3. And the phase 4 is there, but phase 4 is not that much important. Okay, so now in the phase 1 study, we study in very less people like 10 to 10 people, 10 to 100 people. Okay. In phase 2, 100 to 1000. Then in phase 3, more than 1000 patients we have studied. Okay. Then after the clinical trial process, if the clinical trial process gone smoothly and we find out that the drug molecule has effectiveness and it is very much useful, so we go for the approval process, the drug approval, which is given by the US FDA, that is the Food Drug Administration. Okay, so after the drug get approved, it is used for the human mankind, and in the phase 4 study, we perform the what are the side effect of the drug molecule, or you can say long-term side effect. If the person is taking that drug molecule that has been invented, what is the long term? In the long term, what is the side effect that may arise? Okay, so this is the pipeline of drug discovery and drug development. Okay, so now you can see here the unmet need of ADMET. So why we need the ADMET? Okay, so based on the different databases, so we have a target. Then for the target, there are various softwares, or you can say search engines are there, like the drug bank. We have the drug bank. Then we have PubChem. Then we have another tool that is called your Zinc database. Then we have the here it is written Maybreeze. Okay, so all these softwares or search engine are nothing but it is the source of the chemical compound. We can find out all the chemical description, all the information about the chemical compounds we have. Okay, we can find out all the information. So for our target molecule, we need some search engines to find out that what are the possible drug candidate we can take. Okay, so from these 50 million compounds that is present in these softwares, we take 2000 hit molecules. That is the ligand-based virtual screening. So by using the process of ligand-based virtual screening process, we find out 2000 hit, or sometime it may be 100 hits, 1000 number of hits. After that, we perform the molecular docking to screen off the drugs that is not matching the need. Okay. After that, we analyze that molecular docking drugs. Okay, we analyze the results. We analyze the hydrogen bond. We analyze the hydrophobicity. All those things after analyze, we find out that the six molecules are capable of going further. Then after the analysis, we go for the ADMET analysis. Okay, then the bioactivity, all those things we analyze. And finally, we have two heat, or we can say two lead molecules. And after that, these two lead molecule undergoes the DFT process, the molecular dynamic simulation process. Okay, that is the optimization process. So I hope that you get cleared about what is the this process, complete process, entire process of heat identification to lead optimization. So let me summarize this one. So first we have a target. For the target, we have some databases where we can collect the drug molecules like drug bank, PubChem, Zinc. Okay. So from these three databases, we can collect many some sort of ligand molecules. Okay, we can extract, or we can hypothetically design our drug molecule. After that, we perform the molecular docking, and the drug with less binding affinity is get removed. Means it get okay. Then after that, we screen from the 100 of molecules to 20 molecules, because we have screen all those compounds having less binding affinity. Okay. After the molecular docking process, we analyze the interaction. We analyze how much amino acid get binding to the drug molecule. Okay, in this way, we analyze the binding mode analysis. After that, we from the molecular docking drug to after binding analysis, the drug again gets screened, and from the 20, we get six. That's the six hits. Then after that, we analyze the ADMET properties of the drug molecules, the six drug molecules. Then we go for the after we find out that from this six drug molecule ADMET analysis, we find out that two to three compounds are matching properly of all the criteria. It's filling all the criteria. Then we go for the ligand optimization. So this is the process from the heat identification to the lead optimization. I hope that this clears all your doubt about the difference between heat identification to lead optimization. Okay, so I am talking about the process that is the ligand molecule. First, we select the ligand molecule, then we perform the process of docking by using it is also called as the virtual screening process. So we have a hundreds of library, or you can say hundreds of compound. After docking, we have 20 compounds or 30 compounds. Then we go for the pharmacophore modeling. That is, after that we go for pharmacophore modeling, or you can say analysis, all those things. After analysis, we get only 10 compounds. Then we perform the ADMET study, and during the ADMET study, we have again screen the compounds, and we finally have two to three lead molecules. And before that, all these are the hits. Okay, in this process, this is the workflow of your drug design process from the heat identification to lead optimization. Okay. So you can see this plot and you can also find out that how the process is going. Okay. So now next. So I am talking about ADMET, ADMET. So what is this ADMET? So the ADMET is nothing but your first one. A stands for absorption. The D stands here distribution. The M stands for the metabolism, and the E stands for the excretion. Okay. So ADMET is nothing but it is your pharmacokinetic parameter. Okay. So what drug does to our body? When we take any some sort of medication, first what happens? The drug gets disintegrated. Okay. Sorry. The first the drug goes to the body, it gets dissolved. Then after it get dissolved, it get disintegrated, means break down into small granules. Then after that, it goes to the systemic circulation and get distributed to whole of the body part. It get distributed and it enters into the small intestine. Inside the small intestine, we have the microvilli. So through the microvilli, it get distributed, and after that, the drug get metabolized. Okay, so the drug get metabolism, and finally, after showing exerting its activity, it goes for the excretion. So the process of excretion, maybe it is via defecation process, urinary process, or it may sometimes many drug comes from the sweating. Okay, so the excretion process are many types like defecation, or it may be the process of sweating, it may be the process of urination. Okay, you can see that in vitamin capsules. Okay, if we take the consideration of vitamin capsules, so there are those vitamin capsules you can say that in urination they shows red color, and that is the process of they are excreting by the process of this. Okay, okay. So this is the ADMET. ADMET stands for, and the T stands here. The T stands for the toxicity profiling studies. Okay, so this is the toxicity studies. Okay. So now next go to what are the various tools to determine the ADMET, the main portion. So if we have some sort of drug compounds, So how to analyze them? Okay. So what are the tools that we use for the process of ADMET? Okay. So the first tool and the most widely used tool, that is your Swiss ADME. Okay. Then we have the Protox, PKCSM, and finally the path predictor. So in today's session, we are going to demonstrate the Swiss ADME. Then we will demonstrate another AI assisted tool. Okay. So the Swiss ADME is not the AI assisted tool, and we will go for the AI tool, that is ADMET Lab 2.0. Okay. After that, this is the table I want to show you, that what is the difference between the heat and a lead. Okay. So definition of heat. So heat, it is the initial compound showing biological activity against the target. But what is a lead? It is the optimized compound derived from the heat with drug-like potential. Okay. So in the case of heat, that there is might possible of any biological activity or may not, or if it is possible any biological activity is present, that may have the lower potency. Okay. But in case of the lead, after it gets screened many times, it have the higher potency towards the target. Okay. So what are the source for the heat identification? So the heat identification can be done by the high throughput screening process, the virtual screening, docking. So the HTS, the high throughput screening, this process basically, it is a robotic process. It's a robotics process in which millions of compound or more than thousand compounds screen at a time. But in small scale that we use in academics, we use 10 to 20 or max to max 50 compounds. We perform the docking manually and we screen them properly by using the process of docking. We get some results like -7, -8, -9. Based on that, we differentiate or we remove the drug molecule. You can say screen the drug molecules. Okay. Talking about the toxicity risk, the toxicity of heat molecules are unknown because we have a huge set of library, huge compounds. So to analyze each and every drug's toxicity is impossible. So after we perform the docking process, we find out the small amount of heat, then the lead molecule, then we go for toxicity profiling studies. Okay, so number of compounds, if many heats are identified, then developmental stage, it is the early developmental stage. Okay, because we in the early process we find out the heat, then after the all those docking analysis, all those things, we go for the preclinical studies. Okay, after preclinical discovery, we find out that this is our lead molecule. Okay, so the drug development potential is very less and it is having high. Okay, so I have discussed about the heat and lead molecule in a brief way. So now let's go to our second thing, that which I have told you, the tools for the ADMET. So let's analyze by using the process of Swiss ADME. And here I will take the drug for the analysis, that is the amoxicillin. Okay, we'll use amoxicillin for the purpose of the ADMET study, then also for the pharmacophore modeling studies. Okay, so let's start. I will keep these things. So you can see that this is my interface. Okay, let's go to the Swiss ADME. So you have to go to, you can go for Chrome or any search engine, or you can go this as well and search Swiss ADME. I have searched it before, the Swiss ADME. Okay, so go to the Swiss ADME interface. Okay, okay. So Swiss ADME are also known as Swiss drug design. So how to upload your drug molecule? So in here you can see that this in this interface you can draw your drug molecule, or if possible, if you don't want to draw the drug molecule, just simply go here to the PubChem, search the PubChem, click on that, then search your drug molecule, that is amoxicillin, amoxicillin, and you can see here that amoxicillin has been appeared here. Okay, so now click on amoxicillin and you can find out the 2D and 3D structures of the drug molecule. And in this PubChem database, it is just like heaven for the chemistry students, because in the PubChem you can find out all the information of a drug molecule, starting from the lab hazards to the spectral information, all those things you can find out. Okay, so as we are opening the PubChem, let me simply demonstrate the PubChem as well. So how you can find out. Okay, so the first thing, the structures, you can find out the 2D and 3D structure. Talking about the drug molecules lab hazard, so this amoxicillin is irritant in nature and it is having the health hazards. Okay. Then the molecular formula you can find out here, that is for the amoxicillin it is C16H19N3O5S. Okay. Then there are some synonyms are there for the amoxicillin. Now the molecular weight. Okay. So this is the molecular weight of the compound. Then some description, the therapeutic uses, all these things are written. So amoxicillin is a penicillin in which substitution at sixth position, penumbring. Okay. Then after that, you can see that this is the structure. It is containing the beta lactam moiety. Okay. So the four-member ring containing nitrogen and one C double bond O group. It is the beta lactam ring. Then we have the 3D conformer here. Okay, so this is your 3D conformation and they are present in XYZ coordinates. Okay, so if you want to download this one, download the drug molecule, you can just simply go to the option of download here and you can download the 3D HTF format. Okay, so I have downloaded that one also. Then we have the IUPAC name written here. This is the IUPAC name. The INCHL code is given and it is very much important for us. And finally, here we have the smiles. So smiles are nothing but the simple text representation of the complex drug molecule. We have any some sort of drug molecule. So to represent a drug molecule, we have a linear simplified version, that is the smiles. Okay, so these are the text representation of the drug molecules. Okay. Then there we have the cache number. So cache number for the industrial purpose. Okay. So these are some of the things that you can go through. So okay. So for the process of the analysis using the Swiss ADME, what we want to do, we simply just copy the smile here, then go to Swiss ADME. You can paste your smile here. Okay. So if you have designed a new drug molecule, we are unable to provide the smiles. So in that case, what you have to do, you have to draw your ligand molecule by using ChemSketch or ChemDraw. Then after, save into the HTF file format. So as we have downloaded the process, downloaded the drug molecule, we can also open this one in here. Okay, so you can see that we have downloaded the amoxicillin. It is now open here. Okay, so in this process, you can also open the drug molecule by okay. So you have to go to here the option input option, and you can see that there are what are the input files you can provide. the HTF format is here. Then we have the other formats like MDL format is here, mole files format. Okay, the PDB is here. No, the PDB, yeah, PDB data bank format is here also. So in this process, so you can simply enter the smile here, or you can go to the import and you can import your structure here. Okay, so after that, you have to click on run and it will run and it will take some time. Then we can analyze all the properties of the drug molecule. Okay, so here the drug molecule amoxicillin. Okay, now you can see that here completely. This is your amoxicillin drug structure, and this is the biological radar. Okay, bioradar we can say. Okay, where the information about all these things are given: polarity, insolubility, insaturation, flexibility, lipophilicity, and the size. Okay, so based on our smile, we have generated our drug molecule. And talking about now, this is the physiochemical properties. Okay, so talking about the physiochemical property, this is the molecular formula that is given. Then we have the molecular weight, that is a C365.40, number of heavy atoms. Okay, so here comes a tweak, that what is heavy atoms. I hope that you guys are writing all those things from the process of heat lead. Then now next this one, heavy atoms. So heavy atoms are nothing but all the elements that is present apart from hydrogen, that is considered as the heavy atom. So if you can see here in the molecular formula, we have C16, H19, N3O5S. Okay, so the total number of heavy atom we can calculate 16, exclude the hydrogen 19. So 16 carbon, 3 nitrogen, that is the 19, 5 oxygen, that is 24, and 1 sulfur, that is 25. Okay, so in this way, you can calculate the number of heavy atoms. Okay, C16H19. Okay, so now talking about that rotatable bonds. So here we have 5 different rotatable bonds are there. So number of hydrogen bond accepted. Okay, so here the number of hydrogen bond accepted is 6 and number of hydrogen bond donor is 4. Talking about the topological polar surface area. So the topological polar surface area, that is the polar surface present of the drug molecule. What are the polar surface, polar regions? So the polar region is 158.26 angstrom. Okay, and the topological polar surface area must be in between 150. Somehow it is violating topological polar surface area. Okay, so now next talking about the lipophilicity value. So the lipophilicity is denoted by the log P. So here the log P is 0.95. That means it is very less lipophilic. More the lipophilicity value, more is the chance of the drug to cross the blood-brain barrier. Okay, so the lipophilicity value may from the 0 to 5. The 5 is the highest lipophilicity value. Okay, so here in this case, the drug having the lipophilicity value of 0.95. Okay, that is very less. So now talking about the water solubility, it is having the log S value of minus 0.70, and it comes under the class of very soluble. Okay, okay, so you can see here that the drug is having very less log P value, which means it is hydrophilic in nature. So here we have the, it comes under the very soluble class. Then we have the log S another. So these are the various other parameters are there. Okay. So here we have the log S value of minus 0.81. So this parameter of log S, it comes from various journals, various research. So you can see that this is published by in the Journal of Chemical Information Model, Informative Model. So according to that, the water solubility has been taken. So the log S, it is taken from the same journal. Okay, so minus 0.81. So talking about pharmacokinetic parameters. So this drug is having very less pharmacokinetic property, GI absorption. Okay, the GI absorption is very less. It unable to cross the blood-brain barrier. Okay, so the talking about the blood-brain barrier penetration, it is very less penetrating. Then these are the metabolism enzymes. The paraglycoprotein substrate, it also does not inhibit. Here the no means it does not inhibit any some sort of enzymatic activity. Okay, so the enzymatic activity has a very much impact in our body. Okay, so the main cytochrome P enzyme, the CYP here, it stands for the cytochrome P. Okay, so the cytochrome P enzymes have very much important role in our body to metabolize the drug, to metabolize the active drug to the inactive form so that it can excrete out outside the body. From inside the body, it can excrete out. Okay, so it does not inhibit any some sort of metabolism enzymes. Okay, it does not inhibit the cytochrome P2C19 and 23A4. These two enzymes are very much important in our body. Okay, so it does not inhibit them. Then the log K value, the skin penetrate permeation minus 9.94. Okay, so that is also in the same range. Okay, it is not that much skin penetrating activity. Okay, so talking about the Lipinski rule of phi. So Lipinski rule, it is having, you can see here that the molecular weight should be less than 500. It is following that because it have the molecular weight of 365. Log p value less than 5. Then number of hydrogen bond donor, acceptor, all the things having less than 10 and less than 5. Okay, so you can see that acceptor is less than 6. Okay, here it is the number of hydrogen bond acceptor is 6 and donor is 5. So according to Lipinski, it must have less than 4 and less than 6. Okay, less than 5 and less than 6. And it is following all the parameters. TPSA, then donor, acceptor, then the molecular weight, okay, and the log P value. All those things are under acceptable range. Okay, so the drug is this moiety is having the drug likeliness parameter. It is does not follow up violating any some sort of Lipinski rule. So it is following the Lipinski rule, and this Lipinski rule is it is used for the fire. It is also called as the Pfizer filter because it is used by the company that is called as the Pfizer. Okay, then the ghost parameter, it is also have some of sort of like just like Lipinski, it is also have different parameters. So molecular weight should be less than 480. Log P value should be less than equal to 5.6. These two parameters are satisfying our drug molecules. Okay. Then we have waiver. So the waiver is used by the GSK. Okay. So GSK parameter is also called as GSK parameter. Okay. So in this way, you can find out if your desired or the hypothetically designed drug molecule having the drug likeness property or not. Okay. Finally, the bioavailability score. Okay. The bioavailability is nothing but if we take any drug molecule inside the body, it goes and how much the drug molecule from the entire drug is going to the systemic circulation that is called as bioavailability. Okay. Then we have the pains. Okay. The pains are nothing but it is the pan assay interferent compound. Okay. Means there are some drugs like curcumin. You all guys know the curcumin. It is having very much amount of some amount of the properties like it is having anti-cancer activity, it is having the skin penetration activity, it is having anti-tumor activity, all it is very widespread activity, this drug curcumin. Okay, you can find out about curcumin also. So here the pains are not there, it is having zero alert because this drug molecule is not having broad set of activity. If it is having broad set of activity, then it will be showing that one alert or two alert because okay, because what is the needed? What we need? we need zero alert because we want to work on one specific disease. Okay. If here it is showing one alert, we have to find out what is that one alert. That one alert may be anti-tumor or it may be anti-ulcer. Okay. So in this process, we can find out the pains. And finally, the synthesis accessibility is 4.17. Okay. So the more the synthesis accessibility, more is the difficult to design that drug molecule. You can see the synthesis accessibility score here from 1. Okay. So 1 is the very easy to synthesize and 10 is very difficult. And I will tell you that all these things were hypothetical. Okay. If it is AI assisted or it is website, this comes out of softwares. They are just assuming. Okay. They are assuming that the synthesis accessibility is very high. Okay. So don't completely rely on the AI tools. All these tools are only for the education purpose and to awareness. These AI tools are accurate, but you cannot completely blindly use the AI tools for your analysis. Okay. Okay. So this is all about your Swiss ADME. Okay. So this Swiss ADME, it is also widely used. And here you can see there is another thing, the boiled egg. Okay, so you can see that the drug molecule has present here. Okay, so in the boiled egg format, you can see that it is looking like a egg yolk, and this is the white portion of the egg. Okay, so what is happening here? So this is area, the yellow area represents the blood-brain barrier. If your drug is crossing the blood-brain barrier or not. Okay. So now you can see that the drug is not crossing the blood-brain barrier. So the drug molecule, the molecule one is showing here. Okay. So what is why this drug molecule is present here? So you can see that we have the topological polar surface area of 158.26. So accordingly, the drug molecule is present here. If we have a very less topological polar surface area, that drug may present here only or here only, or if we have any some sort of drug molecule that is having the anti-epileptic activity or sedatives, hypnotic drug, anti-anxiety drug, so that drug need to reach the blood-brain barrier. It must need to reach the blood-brain barrier to show its activity. Okay. So for that, if we have any some sort of drug like that, the drug molecule will be present here only inside this yellow portion, or you can say the blood-brain barrier. So here it is also mentioned that BBB stands for the blood-brain barrier. Okay, the PGP, that is the paraglycoprotein. Is it inhibiting the paraglycoprotein enzyme or not? So you can see that the red circle represents that it does not inhibit any paraglycoprotein enzyme. Okay. This is the most important enzyme inside our body, apart from the cytochrome P enzymes. Okay. So this is about the Swiss ADME, and you can retrieve your data in the Excel sheet format. Just click on CSV and your data will be downloaded. So you can see that my data has been downloaded here. Okay. So in this process, you can do the analysis. Okay. So now talking about the publication quality, because everyone needs some publication if working on the in silico process. So you can, as it is a AI session, I don't want to uh, force you that you use the AI, but to analyze the information, you can go to any some sort of AI tool like ChatGPT, Perplexity, and go to Gemini and put this screenshot, and it will analyze for the research paper section. Okay, the section for the research paper, it will give you paragraph. Okay, so that depends on your command, what you are going to command. That you must give a command that convert into a publication quality paragraph, and accordingly it will convert. Okay, we will go to get a demonstration of this one. Okay, so we have completed the ADMET process and we have completed the tools for the ADMET. So now let's go to the, we have learned about the ADME only here. So for the toxicity profiling study, let's go to another software, that is PKCSM or ADMET lab. So I will prefer the ADMET lab 2.0. Okay. So you can see here, this is the interface of ADMET lab. Okay. So this is the interface of ADMET lab. Okay. So here you can analyze the toxicity profiling studies. Okay. So first of all, you have to choose a file in the form of HDF format, or you can enter your smiles here. Okay. You can enter the smile, or you can select the molecule, browse the molecule from the HDF format. So let's go by select the molecular file. Okay. So for that, you have to go to browse and select the compound that we have already downloaded, that is the amoxicillin. Click on open and submit. It will take some time and it will give you the proper results. Okay. So now you can see we have given number of molecule 1, and this is the smile of the drug molecule, and this is the 2D representation. Okay. So now go to the view and it will show you the drug molecule information. See. So in this lab, ADMET lab, you can find out that this is your bioradar, where it is given that the first talking about here, the molecular weight, then these are some of the hate atoms that is present, ring size, TPSA, hydrogen bond donor, acceptor, log D value, log S value, log P value, the log P value, lipophilicity, log S value. It is the water solubility parameter, all the things that has been given. Okay. So here you can see that it is accepted by the Lipinski rule. It is accepted the Pfizer rule, GSK rule, that is the Weber parameter. It is following the Weber parameter also. Golden triangle and the panes. Okay. So if you want to know about that, what is this thing? Here it is in the I option, it is given that what is QED. So the QED is the measure of drug likeliness based on the concept of desirability, whether it is completely attractive drug or unattractive drug. Okay. Or if the drug is too complex. Okay. So you can see here different sections has been given. So this drug comes under the unattractive drug, that is 0.55. But this drug, it is completely used in market for the as a antibiotics for fever or flu. But here it is given as an unattractive section. Okay. So don't rely on the AI tools much more. Okay. Then we have the absorption parameter. It is a Kako cell line. These are the cell line permeability. If in our body cells are present, so what is the permeability to enter inside the cell? Okay, that is minus 6.2, which is the optimal range should be higher than minus 5.15. So it can cross the cell membrane and show its activity. Then there is MDCK permeability. There are three different permeability regions: low permeability, medium permeability, and high permeability. Okay. So this drug comes under the class of medium permeability. Okay. Then paraglycoprotein inhibitor, it inhibitor class G category 0, non-inhibitor. The output value is probably being paraglycoprotein inhibitor. Okay. So in this way you can find out all the information. This is the metabolism information. that has been given. Okay. And you can see that for the clarification endpoint, the prediction probability values are transformed into the six symbols. Okay. So the 3 minus represents 0 to 0.1. Okay. So it does not inhibit, it comes under the category 0 and it does not inhibit any cytochrome P enzymes. Okay, you can see, but it inhibits the cytochrome P2C9. Okay. Now talking about the clearance, it is given that is 2.13 and T half. Okay. T half is given by 0.9. Now talking about the toxicity, this is the main point. We are analyzing the toxicity here. HERG blockers. Okay. Then we have the different properties. Acute oral toxicity. It is don't have any oral toxicity. Then the skin sensitization. It is also don't have any skin sensitization property. It does not have any carcinogenic activity. It does not have any eye corrosion activity. It is not irritant to eye, respiratory toxicity. So you can go through this one, all these things. If you want to download this one, here there is no option of download in the CSV format, but here it is. Okay, so you can download it as a CSV format in the Excel set. Okay, so in this way, we can analyze the ADME by using the Swiss ADME, and you can analyze your drug molecule, the toxicity study, by using the ADMET Lab 2.0. Apart from that, there is PKCSM is there, so you can demonstrate the PKCSM as well. Here in PKCSM, we simply go to PKCSM and the same process we have to do. Okay, so here it is PKCSM. It is getting downloaded, opening. Okay, so you can see this is the interface of PKCSM. So it is a paper that is worked by different organizations, that is the University of Cambridge and the University of Melbourne. They both collaborated to prepare this one. And this work has been published in the Journal of Medicinal Chemistry. Okay, so these are the authors. So they have developed this PKCSM pharmacokinetic parameter profile study tool. Okay, so what you have to do, if you want to predict something, then you can see here there is the option given small molecule pharmacokinetic prediction. So go to PKCSM, click on that, then you can upload the smiles here or you can provide the smiles here. So as we have using the drug amoxicillin, so just copy the smiles here and go to PKCSM and paste it that and click on enter. Okay. So you can see it will showing the absorption parameter and it is currently running. Okay. We can back and we can click on it. We can see that there is an option of absorption, distribution, metabolism, excretion, toxicity. And if you want individual only absorption, then you can just click on absorption or click on distribution. And if you want the whole ADMET prediction study, just simply click on ADMET and it will give you the results. Okay. And I am repeatedly saying that AI tools. It is you can use this for your research paper. You can use just for the knowledge, or you can use for your projects. In your final year project, you can use these tools. But don't use the drug molecule that you have synthesized before going to in vitro studies, before complete optimization. Okay, before validation, you don't use that. Okay, because we have some incidents like this before, so that's why I'm just saying again and again and again. And also, if you have any doubts, you can write down in the chat section and I will reply instantly. Okay. Okay, so now you can see that let's go one by one. Water solubility. It is having very water solubility minus 2.42. Okay, minus 2.42. It is highly water soluble. Then the cell line permeability. Yeah, it is permeability to cell line. Then the intestinal absorption. Okay, it is having high intestinal absorption. High, not high. It is low intestinal absorption. That is 37.35. And we have seen that in the GI absorption in the Swiss ADME. Okay, so in Swiss ADME, we have seen that it is having the GI absorption very low. You can see here, GI absorption is low. And here it is given the same data in PKCSM. Okay, then the skin permeability, it is having minus 2.37 skin permeability. Then paraglycoprotein substrate, yes, it is paraglycoprotein inhibitor. No, okay. Then we have the blood-brain permeability axis. It is not that much blood-brain permeability property. Okay, so this water solubility to paraglycoprotein inhibitor comes under the class of absorption. Then we have the distribution, CNS permeability, blood-brain barrier permeability. Okay, then we have the metabolism. So these are the cytochrome P enzymes that are used for the present inside our body, and it is used for the analysis here. Okay, so now talking about the clearance in excretion portion. So it have no renal clearance and total clearance is zero point 0.3. So toxicity profiling studies, you can see that it is max tolerated dose is 0.17. Okay, and other minor minotoxicity na 4.9. Skin sensitization, there is no skin sensitization, but it is having the important parameter that is called as the hepatotoxicity. So it is having the liver toxicity effect. Okay, so this is all about your softwares that we want to learn. So we have learned about PKCSM, we have learned about the Swiss ADME, we have learned about the ADMET lab 2.0. So you can go through that. You select go to PubChem, then search any drug compound, find the smiles, and you can also prepare your own paragraph for the toxicity profiling studies. Okay, so now next go to our pharmacophore modeling process. Okay, so we have discussed all the things of ADMET. So now AI based pharmacophore model. So before going to the process of pharmacophore modeling, let me show you that what is a pharmacophore. Okay, so we must have to know that what is pharmacophore. So here you can see that we have a pyrimidine ring. Okay, so now this pyrimidine ring has been substituted with various functional groups, like here it is substituted with thiophene, and here it is substituted 2,4-dichloro, here it is substituted with NH2 group. Okay, in this way it became an antimicrobial agent. Okay, this pyrimidine ring has been substituted with this process. Okay, 2, 3, 4, 5 trimethoxy group has been attached. Okay, 3 methoxy groups are present with the benzene ring, and it is having the antioxidant agent. So the main core moiety present here, it is the pyrimidine ring, and this is the pharmacophore. So the pharmacophore, it is the main moiety that is present, main moiety. Okay, so the main moiety here, it is the pyrimidine, and this main moiety is responsible for the activity. As we have seen in the case of amoxicillin, the main moiety that is present is the beta-lactam moiety. Okay, so talking about the antiviral, anti-leishmaniasis, anti-cancer, so you can see that the main core moiety, that is the pyrimidine, is fixed, but there is changes in the substituents takes place. So you can see here the piperazine ring is attached for the anti-malarial activity. Okay, so here the for the antioxidant, as I have told you, that 3-methoxy group containing the benzene ring has been attached. Okay, so here also it is a chalcone. You can see this is the chalcone, and the it is responsible for the anti-leishmaniasis process. Okay, act as the anti-leishmaniasis. So for the AI-based pharmacophore modeling, we have a tool that is called as the DeepFrag. Okay, DeepFrag. Okay, so we can go through that. And if you want such publication quality figure, then you can draw like that by using the process of DFT. So DFT, it is nothing but the study of the difference between the HOMO and LUMO. Okay, you can see the HOMO, that is a higher occupied molecular orbital, and LUMO stands for the lower unoccupied molecular orbital. And between the HOMO and LUMO, what is the energy gap that we calculate by using the process of density functional theory or DFT. And for that, we have so much softwares are present. But now let's focus on the pharmacophore model. Okay. So by the process of before going to the deep frag, let me tell you that how the deep frag works. Okay. So you can see that here we have a benzene ring containing the amide. Okay. So when we attach here the OH group here is attached, it is converted to a drug that is called as the aspirin, or sorry, it is paracetamol. Okay, you can see that because it is contain the OH group, paracetamol, paracetamol, acetyl group. So it's the acetyl group, and mol, it's OL. This is the suffix for the hydroxy. Okay, so this drug, it is your paracetamol. Okay, so now here you can see that this benzene ring is responsible for the analgesic activity, and this hydroxy group is somehow is also responsible for the activity to reduce the flu. So what we do, we have a moiety, then we attach the OH group here, and we find out that this is the activity, the needed activity. Okay, so in the pharmacophore modeling, by using the deep frag, we add the fragments. We just predict the fragment that when we upload your compound molecule or your drug molecule inside the deep frag, it will show that instead of OH group, what are the other groups that may be attached or may be attached here or here. You have to select the position you want to make the change. Then accordingly, it will give the set of result where you can find out that yeah, my drug molecule, I can change this position, I can use the provided substituent for my activity studies. Okay, so I just give you an overall example how the deep frag works. Okay, so we will be briefly demonstrate the deep frag now. Let's keep it here. Okay, so now let's go to the deep frag. I hope that you guys are learning very quickly and you are getting understand all these things. If you are understanding all these things, just spam that yes, sir. Okay. So let's go to, so this is developed by the Durant lab, the deep frag. Okay. So this is the deep frag is a deep convolutional neural network that guides the ligand optimization by extending a ligand molecule fragment. So here in this section, you can see that this is the fragment. This benzene ring is a fragment, and it is a fragment. So we are going to chase any some sort of fragment. We can select the drug molecule, one fragment, and we can see that what are the chains might possible to get a result. Okay, so let's go. So click when open this interface to Durant Lab and click on web application, and it will take you to the site. Okay, input file. Okay. So now we have two input files. We have the receptor and we have the ligand molecule. We have to drop it. Okay. So first of all, we have downloaded the desired drug molecule, which I have earlier told that will work on that drug only. So that is the ligand molecule, amoxicillin. Okay. So before that, we have to find out the receptor. So basically, the amoxicillin, it inhibits the DNA gyrase. Okay, so as I have mentioned that DNA gyrase. So for to download the DNA gyrase, you just simply go to RCSP PDB database. It is the protein database. Okay, so in the protein database, you can find out all the desired protein. Okay, so you can search by the name of the protein, or you can search through the PDB ID if you know. So I have known the PDB ID, that is the 3SRS. Okay, so the 3SRS, it is nothing but the PDB ID of the dihydrofolate reductase. Okay, so you can see that S or S dihydrofolate reductase complex with a quinazoline moiety. Okay, so for the DNA guidance, we can have the PDB ID 1KZN. Okay, so we are going to work on this one. The crystal structure of E. coli domain complex with the chlorobiosine. So this is the DNA guidance, okay, and present in the organism E. coli. So you can see that the DNA guidance interaction is coumarin-based inhibitor. So this is the work that has been published in the biochemistry. Okay, so this is the DOI number. So these are the researchers that they have worked on this, and this is your PDP code. It's written in speak 1KZN. Okay. So in the RCSP PDB, you can find out many different sections like experimental data by using the process of XRD, using the resolution of 2.30 angstrom. This structure of the receptor molecule has been find out that this is the structure of the DNA guidase. Okay. Previously, what I have shown dihydrofolate reductase, that is, we are not going to study that one. Just for the demonstration, I have shown it. Okay. Then this is the submitted work, and this is the ID that is given to this work. Okay. So apart from that, we can see that the DNA guide is having a single chain and having a amino acid sequence rate of 205. Okay. So 205 amino acids are present here, and here the ligand molecule what is attached to the drug receptor, so that is the CBN chlorobiosine is attached. So chlorobiosine is also an antibiotic. So we can compare. Okay. So the chlorobiosine is attached, and this is the 2D representation of the chlorobiosine. Okay. So you can visualize the structure. Let me also show that one, how to visualize the structure of the drug molecule and the receptor. So you can see here in the upper portion, these are the sequence of amino acid as we have the total number of amino acid. So you can see that these are the starting from the glycine. Okay. So we have the amino acid sequence. Okay. So now this is your receptor molecule, and this is your ligand molecule that is present. Let me zoom in. So this is your ligand molecule that is present. That is a co-crystallized ligand molecule. This is called your co-crystallized ligand molecule. And the position where it is located, you can see this is the position where the drug molecule is located. It is also called as the Q site. Okay, so this is called as the Q site, and the reason is called as the pocket atom. Okay, so if I remove the receptor molecule, you can clearly visualize the ligand molecule here. That is the chlorobiosine that is present, and the dotted lines nothing but these are the water molecules. So you can see when I select the the water molecules are appearing. Okay, so all these things you can find out using the PDB access with PDB, and you can see that this is the receptor and this is the drug molecule present. So I'm not going to that deep. Okay, the main purpose is to study the deep frag. Okay, so let's go to the structure summary. So now we can download the 3D representation of the receptor. Okay. So for that we have to go to the download files here, this portion, and found download in the legacy PDF format, and here we have the zip file format also. So first we want to download the PDF format, and you can see that I have downloaded the format. Okay. So now let's go to the different. This is your receptor. So go to the browse and you can see that we have the 1KZ in here. So open and you can see this is the structure. Okay. Okay. So now I have removed the chlorobenzene so that I can test the amoxicillin ligand molecule here. Okay. So there is an option here that remove the co-crystal ligand molecule. So I click on the remove and it has been removed. Now next go to the ligand molecule section. So we can upload the HDF format here. So it is written that formula HDF PDB format we can upload. Okay. So you can see that this is your drug molecule that is present. Okay. And let's go here again to the PubChem, to the amoxicillin and find out that you can see that this is your structure. Okay. So this is your structure of the amoxicillin. So where we want to need to change, we can change the OH group. We can change the NH2 group here, but we cannot change the beta lactam moiety because this is the pharmacophore. Okay. E- Then in the deep frag, we want to find out instead of OH group, what are the other group we attach and we can get a proper activity. All the chemistry students are getting all those things and life science students also, they are taking some time, but they are also getting involved. Means we are substituting this OH group, means we replace this OH group to any other group. Okay. So let's go to deep frag and you can see here that the O group oxygens are represented in yellow color and this the hydrogens in white and the nitrogen in blue. Again the oxygen is white, oxygen is red and the in the gray color we have the carbons. So the sulfur is presented in the yellow color. Okay. So you can see where is the sulfur is present. So here the sulfur is present here at this. Okay. So let's work on it in the defrag. So let's go to here to the defrag and find out where we need the change. So you can see this is your 3D coordinate. It is present in the 3D format and we have to remove the OH group. So you can see that here the benzene ring is present and this one is your OH. So the point you want to change, you just select that one. So I selected the oxygen. Okay, and it is appear in the format of like this that it is selected. Okay, so after you select your fragment from the entire drug molecule, so select it as a select atom as growing point. Okay. Then click on the start deep frag. So it is running right now and it will show instead of the OH group, what is the other group that we may attach to increase activity or increase the binding affinity. So it is currently running. It is an excellent software to find out that instead of this, what we can attach. Okay, so you can see that instead of this OH group, we can attach any some sort of R group. Okay, means methyl we can attach here, ethyl we can attach, then we can attach the alcohol group here. Okay, substituted alcohol. So highest is the activity, highest is the binding affinity. You can see that the out of one the score is been given. So these are the 20 different set of the compound that we might be attached inside of OH group that is present in amoxicillin. So this one. So instead of this OH group, we can attach the R group here. Okay. Yeah. Here. Okay. So now how to analyze this one? So you can see that various groups are there. So first one R, then we have the ethyl hydroxy, That is a substituted hydroxy. We can attach, then we can attach the fluorine, and then we can attach the amines, cyanides. Okay, but the very first rank, that is the score of 1.0, it is the 0 to 1, it is the score. So we can attach this one. Okay. So now go to the files and click on this one, and it will show you. So we can optimize the atomic coordinate and download the smiles. And we can view the fused atom molecule. That is the optimized molecule. So we have clicked on that, and it is currently processing. Okay. So we have downloaded the things, and you can see that this is your ligand molecule smiles. Okay. So we can download in the PDF format as well. And most of the cases we need the PDF format. Okay. So the PDF format has been also downloaded. So you can see that these are the XYZ coordinate of the drug molecules. Okay. So now let's analyze by using the PDF format. So I can go to download and I can open the file. So for the visualization purpose, we use the BioBR Discovery Studio. So now you can see here that here this is the five-member ring that is present, and here the CFC group has been added. You can see that this one, the CFC group. Okay. So here the CFC group is present, and in this process you can do like this. Okay, you can visualize all these things. So by using this DeepFrag software, you can analyze. Okay, so here we have done the changes, that you can see that CFC group has been added here to the benzene ring. Okay, so instead of the OH group, we can add the CFC group. And so what is the purpose of doing all these things? Okay, so now we have the amoxicillin, and we have the hypothetically prepared one drug molecule. So now we can perform the docking and find out what is the difference in the binding energy. It will show the higher activity than the amoxicillin. Okay. Then after we can go for further analysis, ADMET analysis, then go for other analysis as well. So this is the process of pharmacophore modeling by using the AI tool, that is the DeepFrag. Okay. I hope that you guys have understand all these things. Okay. So next, go ahead. So the DeepFrag has been we have learned. Then this is the complete process. Okay. So we have our target receptor, and these are the heat molecules. Okay, so we have a set of heat molecules. Then we perform the docking and we find out that, yeah, this is the ketoconazole structure, and this have been given the some sort of results like this. Okay, so these are the results that has been given, but don't always rely on the result. You have to find out that how much hydrogen bond this drug is forming. Okay. Then after that, we go for the 2D and 3D interaction analysis. You can see that this is the 2D interaction or 2D analysis. Okay, so this is your 2D analysis. Okay, so these are the 2D analysis you can see, and this is a 3D analysis. And these are the bond length has been given. What is the distance between the drug molecule and the amino acid also have been given. Okay, so in this way, we have understand that how to form the pharmacophore modeling using the DeepFrag. Then after that, we have learned about all these things, the ADMET, and then we have discussed the complete overview. This is your target, this is your drug molecules that is present a set of library. Then we perform the docking and find out the best candidate, that is the lead candidate. Okay. So these are the things that we have to learn. So what we have learned today. So we have learned that why most of the drug candidates fail during the process of drug discovery. So the main problem that is ADMET is the main property. Apart from that, other properties that is the less drug likeliness property, less drug likeliness, not following the Lipinski rule of five. Okay, that is the violating the Lipinski property. Okay, next we have the property of synthesis accessibility that we have learned in the suicide ME. Less synthesis accessibility means the higher, easy to synthesize, but if the drug is having more synthesis accessibility, then that drug molecule is hard to synthesize. Okay, so this is why the drug candidates fail. So then we have also learned about how ADMET filters the weak molecules, the weak heat molecules. Okay, at the early stage. Okay. So next we have learned about the pharmacophore modeling and workflow thinking. Okay, don't leave the session because we have some surprise for you guys. We have a surprise for you. Okay, so don't leave the session. We'll consider after the session. Okay, we'll show you. Next, but after all the ADMET analysis, molecular docking process, pharmacophore modeling, such huge things after we do, but why do we get failed? Because we only rely on the tools. We don't understand the chemistry behind the tool, why we are performing this, using these tools. We don't understand the workflow. We do not only rely on the software. You can see that this person is having a score of minus 12.5. He thinks that, yeah, I have get the result of minus 12.5 and I will be rich. I will prepare the drug molecule. I go for the in vitro studies. But here the result is given minus 9.2. But he is analyzing that if my drug molecule is binding to the proper amino acid, is it completely inhibiting the drug molecule or not? Whether my drug molecule is having the synthesis accessibility, all those things. So we have to focus on the synthesis accessibility. We have to focus on the drug likeliness. We don't have to just rely on the tools. The more tools we learn, more smarter we get. That does not happen. We have to we have to learn about the workflow, how to how to analyze the things, how to prepare the drug molecule. So we have weak preparation. We for the docking purpose, we prepare our receptor, we prepare our ligand molecule, but there are some error happens during the preparation, so that it gives a very poor result. So we have to prepare the drug molecule and ligand molecule is for first. Okay, properly we have to prepare. We don't only focus on the score force approach. We don't need this. Binding energy treated as the final result. No, this is not the final result. So these are the things that the student do, the mistake. Okay, so we don't read of the drug, we don't validate the drug molecule, we don't go for the DFT, we skip that during our project or during our thesis writing, or we can say during the publication. Okay, so these are the problems we face while performing the process of the drug design. So we have a surprise for you that to avoid all these things and to overcome all this problem, to get a proper result, we have organized for you a 30 days extensive workshop on the molecular docking. Okay, so where in this process, you will get hands-on molecular docking and ADMET studies. Okay, so you will get hands-on demonstration. Then you will go for the exposure to AI tools and the DFT studies as well. Okay, so you will get hands-on training. Then you get the exposure to DFT and other AI tools. The students who will join, they will be individually guided for the project for the final year project. So many of the BSc, MSc students have been joined in the session, and also many pharma students or biotech students have been joined. So we will give a high research value to for their final year project. Okay, we can give research topics, and it will be individually guided by me. Okay. Next, we'll go from the very scratch, very beginner to advanced level of molecular docking process and publication quality figure if you want. Then we'll go for the publication quality figure. Okay. So research oriented work will done. And it is extensive workshop especially designed for the UG and PG scholars because the PhD scholars have much idea about these things. But the UG students, they are afraid that what is the software, whether I am able to operate the software or not. So that's why we have also designed the workshop in such a way that many UG and PG students will be get benefited. Okay. Next, I will share the complete course module and I will make it transparent to all of you. So we are going to learn all these things in the 30 days workshop in 6 different phases. So first we'll go for the foundation of the drug discovery. The first 5 days will be the foundation. Then we'll learn about how to prepare the ligand molecule and the receptor molecules. We'll learn about extensively about the receptor. We'll go in depth. Okay. Then we'll go for the molecular docking process and the DFT in the next 15 days. Okay. After that, we'll go for the advanced CAD and AI tools that are used. We'll go for that one. Then finally, on the day 20 onwards, we'll go for the project. We'll select the students who need project for their final year. And for those who want the career boost, we'll go for the publication as well. Okay. And finally, we'll publish in the reputed journal. Okay. So now next, go to the workflow. The phase one, you will learn about introduction to drug discovery and drug design. So basics of the medicinal chemistry, how the medicinal chemistry is important, how the quantitative structural activity relationship QSAR is important for the CAD purpose. Then we'll learn the basics of the target, receptor, and the ligand molecules. Then we'll go for the drug discovery pipeline, clinical trials, all those things we will go. Then in the phase two, we'll learn about the how to design a ligand molecule, how to prepare a ligand molecule, then molecular mechanics and DFT studies, and like this we will prepare. Then in the day 10, we'll go for the preparation of the receptor complete SOP, and I will show you 3 to 4 softwares how can you prepare the complete entire receptor. After that, this module has been designed in such a way that you will go systematically. First, learn about the ligand molecule, then learn about their binding homolumo analysis. Then we'll go for the receptor preparation. Then after that, this process of the docking will done. We'll done the multi-ligand docking process will follow as well. Then we can go for the validation and chemistry calculation as well. Okay. So RMST, I will teach you that what is RMST value. Okay. So this in the phase three, we are completely dedicated to the molecular docking process. After that, we'll interpret, analyze all these things. Then pharmacophore modeling, drug repurposing studies we'll also learn. And finally, AI assisted drug discovery we will do. So from the day 20 onwards, we'll go for the publication and mentoring. So for the UG students, we will provide the important research papers or you can say research topics and they can use for their final year project. Okay. And also for their dissertation. Okay. And for the PG students, advanced computational research study molecular docking, ADMET prediction, pharmacophore modeling, DFT, and mechanical insight to the relationship manuscript suitable submission peer review journal. Okay, so this is the perk for your the PG students because we are going to publish those research topics. Okay, in some reputed journals. Okay, so now the thing what you will get in this 30 days program. So from the very scratch to advanced level of molecular docking, you will get to learn. Then high impact research topics will provide you for the final year project or anyone want to publish the paper for their career growth. Then you will get to expose toward the DFT AI tools for the drug design process. Okay. Next, what are the journals we are going to publish. So you can see that these are the journals we are going to publish. So this is the International Journal of Chemical Science, okay, Chemical Studies. Then the Discover Chemistry by the Springer. Then the Oriental Journal. Then there is the Krishnajayati Journal of Applied Biology. Okay, see, these are some of the most reputed journals in which we can publish our paper. Okay, they have some impact factor, and this is very much needed for your career growth if you are any PG scholar, or you can say UG scholar. In UG level, publish the paper is a very much impactful thing, and it impact in your job responsibility as well. Okay, then now talking about the final thing. So we are starting the registration from today onwards. For those who are joining before the February, they are applied for the early bird offer of 2960. So after that, the price will hike up to 3500. Okay. So this is all about from our side. So now the main thing is your response from your side, so that we can proceed further and we'll see you in the live sessions. Okay. So these are the other journals as well, so that the no APC will be charged. We will not take any money from you. We just simply publish your work. Okay. So thank you so much for joining the session. And if you have any doubts, you can write in the comment section or in the chat option. Okay. So thank you so much. See you soon in the live session. Thank you.